oxidative phosphorylation. What does it mean? Let us break down these words first. Oxidative means produced by oxidation. And what is oxidation? It is either gain of oxygen, loss of electron, or loss of proton. Phosphorylation means addition of phosphoryl group. Now where is this phosphoryl group added? It will be added to ADP and form ATP. I will explain the significance of this name later, so do watch out for that. The process of oxidative phosphorylation is basically occurring in the fifth complex of electron transport chain. Let us talk a bit about this complex first. It is also known as ATP synthase. As it is an enzyme, so it will act as a catalyst to aid in production of ATP without being used up. The major structure of this complex contains two portions or two subunits, F0 and F1. F0 portion will serve as proton channel through which proton will be entering mitochondria. It is a disc-shaped structure with division into C subunits. There is then attachment of gamma subunit on its bent axle. This gamma subunit will fit itself inside the F1 complex, now moving towards F1 subunit. Now this F1 projects into mitochondrial matrix. This subunit will basically catalyze ATP synthesis. F1 complex consists of 3 alpha chains and 3 beta chains. Gamma subunit will be fixed inside these 6 chains. Other than that, some studies showed that there is sigma and epsilon subunits as well, but their functions are not quite defined yet. Alright, so to start this process, we are missing 2 molecules. One is ADP molecule and another one is phosphate group. So, where will they come from mitochondria? Now in the introduction to ATP video, we mentioned that is carrier known as ATP ADP translocase, which will transfer ADP molecule here. And what about phosphate group? It will come from phosphate carrier protein present in inner membrane of mitochondria. Now all the components are completed. But how will the process start? 3 major proposals for the mechanism of oxidative phosphorylation have been considered. The synthesis of ATP is carried out by a molecular assembly in the inner mitochondrial membrane, which we name as complex 5. First hypothesis is known as chemical coupling hypothesis, in which there is a concept that there is production of high-energy intermediate after electron transport chain that will undergo this process of production of ATP. This type of intermediate has not been found, so this hypothesis is not much used. Second hypothesis is the conformational coupling hypothesis, in which they proposed that mitochondrial cristae undergo certain changes that will lead towards the process of ATP production. These conformational changes reflect the changes in different components of electron transport chain. So it is also believed that these changes represent the formation of high-energy state. Then comes chemiosmotic theory. This theory is most accepted view of oxidative phosphorylation. Mitchell's chemiosmotic theory postulates that the energy from oxidation of components in the respiration chain is coupled to the translocation of hydrogen ions, or protons, from the inside to the outside of the inner mitochondrial membrane. Each of the respiratory chain complexes 1, 3, and 4 acts as a proton pump. The inner membrane is impermeable to ions in general, but particularly to protons, which accumulate outside the membrane, creating an electrochemical potential difference across the membrane. This consists of a chemical potential difference in pH and an electrical potential. The electrochemical potential resulting from the asymmetric distribution of the hydrogen ion is used to drive the mechanism responsible for the formation of ATP. Alright, so we have established that there is electrochemical potential difference across the membrane that will trigger oxidative phosphorylation and generation of ATP. Let us now understand the main mechanism of ATP production. There is a hypothesis known as Boyer's hypothesis that states the binding change mechanism for ATP production. According to this hypothesis, beta subunits of ATP synthase are responsible for generation of ATP. According to this hypothesis, 3 beta subunits, catalytic sites, though structurally similar but functionally not same at a particular time. It is envisaged that beta subunits occur in 3 forms: O form, L form, and T form. Remember them by a mnemonic known as LOT. O form is also known as open form. This form is known to have low affinity for substrates ADP and inorganic phosphate. The next one is L form. It is also known as loose form. In this form, ADP and inorganic phosphate binds with more affinity than the O form, but catalytically it is inactive. Last one is the T form, which is for tight form, in which substrate ADP and inorganic phosphate binds tightly with this form and catalyzes ATP synthesis. Each form is interconvertible into each other by certain conformational changes. Now this hypothesis is modified by rotary or engine driving model. According to this model, it is now widely accepted that protons passing through the disk of C subunits of F0 subcomplex cause it and the attached gamma subunit to rotate. The beta subunits, which are fixed to membrane, do not rotate. ADP and inorganic phosphate are taken up sequentially by the beta subunits, which undergo conformational changes, and forms ATP, which is expelled as the rotating gamma subunit squeezes each beta subunit in turn. Thus, 3 ATP molecules are generated per revolution, as one revolution will allow 3 beta subunits to rotate. Now, as a whole, to summarize this procedure in a single sentence, is that there will be electrochemical gradient produced when all the protons will come inside intermembrane space. To eliminate this difference, C sides of F0 portion of a TP synthase will move, making a rotation of gamma subunit and ultimately beta subunits. These beta subunits, when come into their tight form, will bind ADP and inorganic phosphate to form ATP molecule. Now, when there is removal of 4 protons, 1 molecule of a TP will be produced. What could be the reason? Protons flow down their concentration gradient into the matrix through the membrane protein ATP synthase, causing it to spin like a water wheel and catalyze conversion of ADP to ATP. So, these protons will help in spinning of this enzyme ATP synthase. So now the name of the process is justified, as there is loss of protons, which is oxidation, and addition of phosphoryl group to ADP. So here is oxidative phosphorylation explained. Keep watching, as we will be discussing respiratory poisons that might affect these processes next.